I haven't had a chance to look at them. I just got them myself, so you know as much as I do about them at this point. Maybe more because you took it at least. So, okay. I want to continue talking about fatty acid oxidation, finish up talking about oxidation of unsaturated fatty acids, and then we'll turn our attention to ketone bodies and fatty acid synthesis. So we probably won't finish fatty acid synthesis today, but we will finish that on Wednesday. And along with that, I'll be talking about prostaglandin metabolism, which is a very interesting aspect of biochemistry as well. I know I say that about all the different pathways, but I do find that an interesting pathway. Okay, well, last time we talked about how saturated fatty acids get oxidized. And one name I didn't give you, I probably should give you. We talked about this type of oxidation, fatty acid oxidation, that I described last time as what's called beta oxidation. So if I can just show you that real briefly. Beta oxidation gets its name, not the one I want, gets its name from the fact that if we number the carbons, okay, there's the alpha carbon, there's the beta carbon. All the action, as I said, is occurring between carbons 2 and 3. So we call it beta oxidation because the oxidation is occurring right here. So beta oxidation simply refers to oxidation of saturated fatty acids. Not all fatty acids in our body or in our diet are saturated, so we have to consider the oxidation of unsaturated fatty acids, and they are a little bit more complicated. I think that they're not as bad as they seem in the sense that you can do everything that we need to with unsaturated fatty acids by the action of 2 enzymes. There's really only 2 enzymes that we need to worry about in oxidizing unsaturated fatty acids. The first thing you see on the screen here. This shows the oxidation of palmitoyl, I can't even say it, CoA. And that's just basically a palmitic acid that has a double bond in it. Palmitic acid has 16 carbons. And fatty acid oxidation starts, that is beta oxidation, starts in the same way. That is, we can go through and we can block or cut off 2 carbon pieces of acetyl-CoA through several rounds of oxidation until we get to this stage right down here. So we've knocked off several of these acetyl-CoAs in the regular rounds, that's shown here by these arrows. And when we get to this point, there's some changes that have to happen. Okay? So at this point, the enzymes that would normally proceed through beta oxidation, specifically the acetyl-CoA dehydrogenase, can't do anything with carbons 2 and 3 because, as you can see, there's a double bond over here between 3 and 4. So when that situation occurs, notice that the double bond is a cis double bond. When that double bond appears in positions 3 and 4, as it would during the oxidation of an unsaturated fatty acid, there's an enzyme called cis-delta 3-enoyl-CoA isomerase. I know, you want to... What's it going to be?
I did have dibs on Virginia.
We had dibs on Virginia. Cletus. Who said that? Cletus. I like Cletus, okay? So I will accept either cis-delta 3-enoyl-CoA isomerase or Cletus as the name of this enzyme. Now, Cletus, and if you spell something reasonably close to Cletus, we will give you credit. Okay, Cletus, I like that. I had a good friend in high school named Cletus Klauser. That was his name, and there's nothing wrong with it. I mean, he was a nice. What's wrong with Cletus? I like the name. I hope there's nobody in here named Cletus. I'll be in trouble. I'm not making fun. I like the name. I really do. Anyway, so Cletus, what he does is catalyzes the movement of this double bond. So it converts a 3,4 cis to a 2,3 trans. That's basically what it does. Now, you remember that a 2,3 trans is a normal intermediate in fatty acid beta oxidation, right? So what would be the next step in the oxidation of this compound? Anybody remember? When we oxidize a fatty acid, the first step is dehydrogenation. What's the next step? Adding water, right? We add water across the double bond. That would put the hydroxyl here, right? And then we would oxidize that hydroxyl to a ketone, and then we would break an acetyl-CoA off, right? So all we have to do is move that double bond from being a cis 3,4 to being a trans 2,3, and we can proceed with beta oxidation just as we normally would. Okay? That makes sense? So Cletus does that. Well, we have another enzyme that the things don't always come up in the 3,4 position. So what if they come up in a different position? How do we deal with them? Well, this figure shows the oxidation of malonyl-CoA. I never can say them. I know I'm not going to give them a name too. We'll have names for everything. Okay. It proceeds through beta oxidation, and it gets to this point here. And in this case, we have a double bond that is a cis 3,4, and it has the Cletus can act on this. And when Cletus acts on this, what's going to happen to the cis 3,4? Well, it's going to become a trans 2,3. And we can go ahead and we can continue with regular beta oxidation right here. And when we do that, we will eventually clip this guy right here. And that will generate this compound down here. Now, this compound is different than what we saw here. Okay? What we see here is we've got a cis double bond at position 1, 2, 3, 4, 5. Okay? This was a cis double bond position 3, 4. So when we have that cis double bond between 4 and 5, we have to go through a couple more steps to be able to oxidize this guy. And they're interesting and unusual. So the first of these starts out just like beta oxidation. Here's our fatty acyl-CoA dehydrogenase. That's the first enzyme in the oxidation pathway. And it removes electrons between carbons 2 and 3, just like we're starting beta oxidation. Okay? So the very first step of this process removes electrons just like we would do in a regular beta oxidation. However, the next step, we've got a pair of double bonds that are together, and that causes some problems for the next enzyme in the pathway. That pair of double bonds has to be dealt with, and it's dealt with a new enzyme. The new enzyme is 2,4-dienoyl-CoA reductase, and I'm open to names. Kevin. I don't think I want to call it Kevin. Wallace. Wallace? What was that? Daryl? Wallace? How many like Wallace? How many like Daryl? I think Wallace has it. Okay. So we have Cletus and we have Wallace. You can call it by the proper name, and yes, we will give you credit. Okay. Here's Wallace. Wallace has an interesting property. Okay. Now Wallace is taking something. Let's look at the big picture here. Shh. The big picture is that we have two double bonds here. They're getting converted into one double bond here. And it's at this point your book is wrong. Okay? Two bonds do get converted to one, but the bond that's made is not trans, it's a cis. Okay? The correct bond is actually a cis bond. Well, how do we convert two double bonds to one? Well, that means we have to add electrons, and we add electrons from NADPH. Okay? Because we're essentially converting two bonds to one. Those electrons have to come from somewhere. They come from NADPH. That gives us a cis, okay, fatty acid that has a cis bond between carbons 3 and 4. 1, 2, 3, 4. Right? Well, that's no different than we saw over here. A cis bond between carbons 1, 2, 3, 4. And what enzyme is going to act on that? Cletus. Okay? So using these two enzymes, we can basically handle all of the normal biological intermediates that we have of cis double bonds that occur in fatty acids. Okay? I didn't say we could handle trans double bonds that might happen in trans fat. And you could imagine there might be some complications with that, and in fact there are. Okay? That may be why we have problems in metabolizing and dealing with trans fats in our diet. The cis double bonds we can handle, and as a consequence, we can oxidize fatty acids that have the cis double bonds all the way down to acetyl-CoAs. Okay? Which is good because we use fatty acids to store energy. Right? One thing I didn't mention about fatty acid oxidation I should is really important relative to energy. Okay? Now all these, of course, depend on how you count your ATPs. But if we take a single palmitic acid and we oxidize it all the way to carbon dioxide, we get about 127 ATPs, depending upon how you count them. One glucose gave us 38. And even if you allow for the difference between the number of carbons in glucose and the number of carbons in palmitate, we still get more ATPs out of palmitate. So fatty acids are a greater reserve of energy for us than glucose is. Now, we don't use fatty acids for the most part for rapid bursts of energy because, as you've seen, why? The body has to package those things up into LDLs and HDLs and VLDLs and all those different components and transport them through the bloodstream. That takes time. And if we need instant energy, that's not going to do us good. On the other hand, the liver can directly dump glucose in the bloodstream and have it be used as an energy source. Julie?
Does Wallace always only produce cis, or do you ever have trans come out of it? Because I would imagine you have free rotation on that alpha carbon.
Yeah. As far as I know, it always produces cis. Yeah. Always produces cis.
That alpha carbon, I mean, aside from sterics, you would have rotation there.
The alpha carbon here?
Yeah.
Yeah. But again, as far as I know, you only get cis out of that because that's what is needed for this guy to go forward. I don't know that you get much trans. I don't know that, Julie. Yes?
Can you follow the double bond all the way through? Because I'm a little confused what happens between 3 and 4 and how these double bonds got one carbon closer overall.
You're talking about the double bonds from here upwards? Is that what you're saying? Or from here?
Yeah, from starting from there, yeah. So 3 and 4, we lose a double bond.
Okay. So we've got several chops up here. You understand how we get to here?
Sure.
Okay. So we get to here, and we've got a cis double bond between carbons 3 and 4, right? That cis double bond between carbons 3 and 4 is moved to a trans of 2,3 by this cleaves enzyme. Okay? So this now can be oxidized by regular beta oxidation. I can take and I can add water across this double bond, put an OH here, make a ketone here, and lop off this acetyl group. That now produces, here's this carbon here. This carbon becomes this carbon, because we put a ketone on there, remember. Now, we've got 1, 2, 3, 4, 5. We've got a cis between 4 and 5. Up here, we had a cis between 3 and 4. Okay? We start the oxidation process here just like it's a beta oxidation. And in a beta oxidation, we would go ahead and we would remove the electrons between carbons 2 and 3, which is what we do. Removing those electrons produces a conjugated set of double bonds here. Conjugated meaning they're separated by one single bond. A conjugated set of double bonds here. And that conjugated set of double bonds can't be handled by the next enzyme of beta oxidation. Okay? So that means, therefore, that we have to fix this, basically. And that's where this enzyme Wallace comes into play. And what it's doing now is it's converting 2 double bonds into 1 single bond. Okay? As I said, this ends up being a cis bond, not a trans bond. And the reaction requires electrons. Electrons coming from NADPH. That puts a cis bond at position 3 and 4. But a cis bond at position 3 and 4 is exactly what this enzyme can handle. So this enzyme converts a cis 3,4 to a trans 2,3. And we go through regular beta oxidation for the rest of it. Does that make sense? Okay. It is a little complicated, but really when you look at it, there's only 2 enzymes that are doing this. They're able to interconvert the forms as necessary so that we can ultimately get to beta oxidation. That's what we're trying to do. We're trying to oxidize these compounds by the regular steps: dehydrogenation, hydration, oxidation, and then splitting off of a CoA, acetyl-CoA. Okay. So if there are no other questions, that's the last of the unsaturated fatty acid oxidation. Yes, Jen.
Can you recap actually what we do with the trans 2-enoyl-CoA once we get there?
what we do with the trans, let's see, the trans,
That last step, like what if you do this?
this guy here? Oh, yeah, sure. Okay, so this is just a regular intermediate in beta oxidation. So again, we would add water to this, because here we've got a trans between positions 2 and 3. We would add water, which would put a hydroxyl there on carbon number 3. We would oxidize that hydroxyl to a ketone, and then we would just simply split off an acetyl-CoA like in regular beta oxidation. That would then give us something that looks like this. Of course, now everything's going to be single bonds, and we just go through the regular oxidation process for that. Okay, make sense?
Yeah.
Yes?
How many acetyl-CoAs are given off?
How many acetyl-CoAs are given off? So it depends, obviously, on the number we start with. So if we start with a fatty acid like linyl-CoA, we start with 18. So we would give off 9 acetyl-CoAs.
Is there any given off between the step below?
From here to here?
From,
Here to here?
yeah, from the molecule before cleavage up to trans 2 enzyme-CoA. Is there any given
From here to...
off all the way through?
Okay, well, you can, okay, so they've color-coded it. You can see this black comes off. So that's lost in going from here down to here. In other words, this is just beta oxidation that's occurring here. All right? So there's one acetyl-CoA that's lost there. All right? No other acetyl-CoAs are lost in the rest of this scheme. Only after you get back up here can you start losing them again. Make sense? So a lot of this is just rearrangement to get the appropriate substrate so that we can go through and do the regular oxidation scheme. Okay? Okay. We're clear on this? Cletus and Wallace. I got to remember that. Wallace and Gromit. We should have Wallace and Gromit. That would have been great, wouldn't it? Oh, well. No, I like Cletus. Let's leave Cletus. Okay. Well, there's one other consideration we have to have for oxidizing fatty acids. You're probably getting tired of oxidizing fatty acids. But there are rare cases where fatty acids have odd numbers of carbons. Okay? Everything we've done so far, we've assumed they had even numbers of carbons. And in fact, by and large, the vast majority of fatty acids do, in fact, have even numbers of fatty acids. So that means every time we chop off 2, we eventually get down to something that has 4. When we chop that in half, we get 2 2s. Okay? Well, what if we have an odd number of carbons? Let's say we had something that had 17 carbons instead of 16 carbons. What's going to happen when I oxidize that? Well, beta oxidation, assuming it's a saturated fatty acid, beta oxidation is going to proceed normally as it would if I had palmitate, which has 16 carbons. The place we run into trouble is when we get down to the last 3 carbons. When we get to that point, the enzymes of beta oxidation won't touch that compound. Okay? They won't touch a 3-carbon intermediate at the very end. So at that point, the body has to do something, or the cell has to do something with handling that 3-carbon piece. And what it does is interesting and it's odd. This is one place where you look at metabolism and you go, what the hell? What were they thinking? Okay? Why does this go through this process? Let me show you what happens. We've got a 3-carbon piece called propionyl-CoA. Okay? Propionyl-CoA, in order to be metabolized in the body, needs to gain a carbon. Okay? So it gains a carbon, and the carbon that it gains is on position, the middle carbon of the 3. So this guy right here gains a carboxyl group. Now, what the body's ultimately wanting to do is make succinyl-CoA. Why doesn't the body put this carboxyl group on the end? Don't have an answer for that question. Okay? Don't have an answer for that question. Metabolism isn't always logical, as you will see. Yes, sir?
Maybe the resonance structures make the carbon easier to attach.
You're right. There are chemical reasons for this, and I'm just arguing this from a philosophical point of view. But you're right. There are chemical reasons for that. Okay? But looking at this, you think, you know, enzymes evolve some pretty cool mechanisms, and we're going to see some of those cool mechanisms. But how comes enzymes, we have not evolved enzymes that handle a very interesting mechanism to do this? Well, it puts a carbon on the middle carbon. Not only does it put a carbon on the middle carbon, it puts a carbon in the middle carbon in a configuration it doesn't like. It puts it on the D configuration, and then it turns around and converts it to an L. Okay? Does that make any sense? Not to me. All right? Why didn't you put it at least in the L configuration to start? Okay? Well, it turns out the enzymes, again, aren't evolved that way, but this moves from the D configuration to the L configuration, and only after it's gotten into the L configuration does a final enzyme catalyze the movement of this carboxyl to the end of the compound. Okay? Now, this is an unusual set of reactions. This produces succinyl-CoA, and this is one of those minor exceptions I told you about where we actually could take a piece of a fatty acid and make it into glucose. How would we make this into glucose? By going through oxaloacetate, right? Through the citric acid cycle. Now we would have an extra oxaloacetate, and we have a very minor way of making glucose using a part of a fatty acid, a part of a fatty acid, that is. Okay? All right. So this is an odd reaction, and the odd part of the reaction is actually this movement of this methyl group swapping positions here. This odd part of this reaction actually requires the enzyme B12. Okay? B12 is necessary for the movement of that, that methyl group swapping positions with the hydrogen. Okay? That B12 looks like this. B12 is a very elaborate, very complicated compound, and the cobalt that you see right there is involved in actually grabbing a hold of that methyl group and facilitating that swap in configuration. So the cobalt plays a very important role in that process. As far as I know, this is the only place in all of biology that cobalt is needed. Okay? So cobalt is an essential nutrient because of this. Vitamin B12 is obviously important. Vitamin B12 does some other things as well, but this is one of the interesting things that vitamin B12 does in the overall process. Okay. Mechanism I won't go through. The last thing I will talk about actually has more to do with cell biology than it does with metabolism. And this part refers to different compartments in the cell in which oxidation is occurring. So far we've only talked about fatty acid oxidation occurring in the mitochondrion, in the matrix of the mitochondrion specifically. Okay? It turns out that fatty acids get oxidized in the mitochondrion in two places. All right? Most of the oxidation occurs in the mitochondrion. But oxidation of long fatty acids starts in the an organelle called the peroxisome. Okay? So if something is longer than 16 carbons, like we have fatty acids, for example, that might have 18 or 20 or 22 or even 24, oxidation of those starts in the peroxisome. Why does it do that? Well, there are reasons that I won't go into here for why the cell does that, but you actually have a hint that there might be some division of labor in the cell with respect to oxidation of fatty acids. If you remember, the very first enzyme in the oxidation of fatty acids was an acetyl-CoA dehydrogenase. That enzyme came in three forms. A form that worked on long fatty acids, a form that worked in medium chain fatty acids, and a form that worked in short fatty acids. The medium one, I said, was the one that was deficient in children who die of sudden infant death syndrome. Okay? It turns out that the one that works on the long fatty acids, is only found in the peroxisome. So the acetyl-CoA dehydrogenase that only works on long fatty acids is found only in the peroxisomes. What you see is an electron micrograph of a peroxisome. Okay, question? No, okay.
What was the length of chain separation?
So basically, if it's longer than about 16, oxidation of that fatty acid will start in the peroxisome. Now, peroxisomes don't have an electron transport system. They do have FAD, they do have NAD, but they don't have an electron transport system. So they've got to do something, they've got to do something with those electrons that get transferred to FAD during oxidation. This shows that those electrons in the peroxisome get transferred to FAD to make FADH2, and then FADH2 donates its electrons to oxygen to make hydrogen peroxide. I started to say water. Okay, it makes hydrogen peroxide. Now, This is interesting, okay, in a couple of senses. All right? One, it looks like we're losing some energy, right? Instead of those electrons going to the electron transport system, some of those electrons are just being made into hydrogen peroxide. So peroxisomal oxidation is not nearly as energy efficient as it is in the mitochondrion. We're losing some electrons being transferred through electron transport, which means we're losing some protons being pumped, which means we're losing some ATPs. Okay? The other thing that's of interest is the peroxisome is involved. It's what people refer to as the recycling engine or the garbage cleaner of the cell. And one of the things that peroxisomes do is they help to oxidize things. This hydrogen peroxide is a reactive oxygen species. This actually may facilitate oxidation reactions occurring on other molecules in the peroxisome. As you can see, it can be broken down, but it can also be used as a reactive oxygen species. So the peroxisome has the benefit of making something, this guy right here, hydrogen peroxide, that it might be able to use for other reactions. Okay? Yes?
So the longer fatty acids just start their oxidation in the peroxisome?
So, good question. So his question is, do they just simply start there and nothing else happens? And the answer is no. They start and they actually chop off a couple of pieces until they get down to about 16, and then they get moved to the mitochondrion. I'm not showing you the rest of the reaction. I'm just showing you the first part. But the peroxisomal oxidation does, in fact, go through the same basic steps that we see in the mitochondria. We see a dehydrogenation, okay, which is occurring right here. Then we're going to add water across this double bond, make an OH, make a ketone, split off an acetyl-CoA. So all of those steps will occur in the peroxisome like they occur in the mitochondrion. But when we do these steps in the peroxisome, we're not, in fact, going to get as many ATPs because we don't have that electron transport system. Okay? Okay. So peroxisomal oxidation is important for the long-chain fatty acids. All right. Let's turn our attention now to a related set of compounds called ketone bodies. And ketone bodies are something that I think most people have heard something about. They're interesting compounds, and they're basically what I like to describe as emergency energy sources for the body. They're emergency energy sources for the body. Now, why do we need emergency energy sources for the body? I'll give you a real good reason. All right? Let's imagine that you are on a starvation diet. Okay? And you're not eating anything at all. What's happening in your body? Well, your body has a constant need for things like glucose. Your brain relies on glucose. Your eyes rely on glucose. People who starve themselves to death usually go blind before they finally die from starvation. Okay? So you need this glucose that's there. You have a constant need for glucose. All right? Well, you can get some of that glucose from breaking down proteins. You start breaking down your muscle proteins, you take those amino acids, and you've seen how you can convert, for example, glutamate into a citric acid cycle intermediate. You can convert aspartic acid into oxaloacetate and make glucose. But you start drawing on your reserves, you don't have anything else left. Okay? You might be diabetic, where you're having trouble managing your levels of blood glucose, in which case your blood glucose levels may go very low at some point. And the body still has a constant need for glucose. All right? Well, for emergency situations, what the body does is it makes something called ketone bodies to supply energy to the brain, primarily to keep the brain going. You don't want to pass out because you're not eating enough. So that does happen as well. How does this work? Well, the way it works is as follows. We start with 2 acetyl-CoA molecules, and using our enzyme thiolase, we'd run that reaction in reverse that we ran in oxidation. So instead of splitting a 4 into 2 twos, we take 2 twos and put them together and make a 4. This is the acetoacetyl-CoA that you saw during the synthesis of cholesterol. Acetoacetyl-CoA, again, like in the synthesis of cholesterol, adds another acetyl-CoA to make a 6-carbon molecule, which is HMG-CoA. And that's, again, the same intermediate that we saw in the pathway to make cholesterol. Now, if we were going in the cholesterol direction, we take HMG-CoA and we make it into mevalonate. That's one branch. If we go that branch, we make cholesterol. If we go the branch that you see here, we make ketone bodies. All right? So if we take HMG-CoA and we pull off an acetyl-CoA, we are left with a molecule of acetoacetate. Acetoacetate is a ketone body. Now, a couple things can happen to acetoacetate. One is I can have chemical breakdown of acetoacetate. If I do, that's not good, because what happens is I make a 3-carbon compound, acetone. Everybody knows what acetone smells like. Acetone is of no use to my body. I exhale it from my lungs. People who are having problems handling glucose, you can smell acetone on their breath. If you have a friend and you can smell acetone on their breath, you should tell them, get to a doctor. There's a good chance they have diabetes. Okay? Yes?
Is that why people that are going in a diabetic shock are sometimes mistaken for being alcoholics?
It can be, because it has an organic smell to it. That may well be the case in terms of being mistaken for that. But this acetone smell is quite prominent, and there probably are people in here who actually have noticed it. If you ever notice this and somebody say, get to the doctor, because you probably are having some issues with glucose metabolism, and that likely is a sign of diabetes. Yes, sir?
I've heard this can happen to people who are on the Atkins diet.
Yeah, you hear these sorts of things about Atkins diet, and in my experience, that is a bit of an exaggeration. Okay? And the reason it's an exaggeration is because in the Atkins diet, what you're getting is plenty of protein. You're still eating. And so in the Atkins diet, you're converting, you still have amino acids that you can convert into glucose. You may have this occur to a very limited extent, but not anything like the extent where you have starvation. So it's a very different kind of phenomenon. The only way this would happen if you were on an Atkins diet would be if you ate only fat. And that's impossible to do, essentially. Yeah. Okay? But it's a very common question, and people do think that happens on Atkins. It's not a big problem in Atkins. All right. Well, I haven't told you about the ketone bodies. So one thing can happen, and this process that goes to acetone is a chemical instability. This is not an enzymatic reaction. Acetoacetate is chemically unstable. It's chemically unstable. So that happens with a certain frequency. Well, the body doesn't want its acetoacetate going to acetone, and then you breathe it out the lungs, because remember, the reason we're making ketone bodies in the first place is so that we can keep the brain alive. We want to get energy to the brain. The other possibility is that the body takes that acetoacetate and it converts it into D3-hydroxybutyrate, which is much more stable. This guy can travel in the bloodstream without a significant amount of breakdown like we saw with acetoacetate itself. Okay? Well, what happens? This guy travels in the bloodstream. It will cross the blood-brain barrier. It gets to the brain, and what does the brain do with it? Well, the brain does the following. The reversal of all those steps, and now it's got 2 acetyl-CoAs. The brain just got some energy, because the brain can use acetyl-CoA in the citric acid cycle for energy. And of course, we know that's a major energy source of the body. So, using ketone bodies helps the brain to stay alive. Another important consideration here is that fatty acids, when broken down, what do we get plenty of with fatty acids? Acetyl-CoA, right? So even though we can't convert acetyl-CoA into glucose, we can use the energy of fatty acids to keep the brain going when we need to. Okay? Make sense? Yes, Jim?
Is the brain the only thing that can do this?
No. But this is important for the brain because the brain needs that constant supply of energy. Yeah. Yes?
So you still have to have the acetyl-CoAs in your body somehow from...
Oh, yes, absolutely. If you don't have a source of this, that's right. That's right. So you're going to be breaking down your fat. You betcha.
Way to convert it, to get it from one place to the brain.
That's right. Yeah. Yes?
Where does the acidosis part come into play? What causes acidosis?
Okay, so what causes acidosis with this? One of the things that you're releasing here is carbon dioxide. And carbon dioxide will be converted to carbonic acid, and that's where you're getting the acids that's coming about. That's one of the ways you're getting it. Okay, good questions. Now let's see. I've got about 10 minutes. So that gets us a chance to get started on fatty acid synthesis. Now, fatty acid synthesis, when I showed you the sort of the chemical reactions necessary for making a fatty acid, breaking a fatty acid down, I said that the synthesis was chemically very similar to the reverse. I said there were some exceptions, and I'm going to take you through those exceptions. But in general, the chemistry of this overall process is not unlike the reverse of fatty acid oxidation. One of the places where you see a difference is actually on the screen right here. This is an odd step, again, that the cell takes in order to make a fatty acid. To make fatty acids, cells start with a 2-carbon molecule of acetyl-CoA. Okay? Acetyl-CoA gains a carboxyl group in the process. Okay? So we're making an intermediate called malonyl-CoA, which has 3 carbons. Now what we're going to see is that the cell actually chops off that carboxyl group it put on in the process of growing the fatty acid chain. So this is only on here transiently, but this is an important step in the synthesis of fatty acids. This reaction that you see on the screen is catalyzed by the only regulated enzyme in fatty acid synthesis. The only regulated enzyme in fatty acid synthesis. It's called acetyl-CoA carboxylase. And no, we won't have a different name for that. Okay. Acetyl-CoA carboxylase. Acetyl-CoA carboxylase catalyzes this reaction. And as I said, it's the only regulated enzyme in fatty acid synthesis. Its regulation is interesting and complicated. So before I show you the reactions of synthesis, I'm going to show you how this enzyme is regulated. It skips down to here, unfortunately. Okay. First of all, acetyl-CoA carboxylase can exist as both a monomer and in chains of polymers. You can actually have individual units, link, make very long polymers in the cell. And what you see here are electron micrographs of polymers of acetyl-CoA carboxylase. All right? The polymers are the most active form of the enzyme. Okay? They're the most active form of the enzyme. The polymers are in a dephosphorylated state. They do not have phosphate on them. Okay? Phosphorylation of individual monomers will cause the polymer to fall apart and leave the enzyme in a less active state. So dephosphorylation makes it active. Phosphorylation makes it relatively inactive. Okay? Well, you can probably guess what enzymes might be involved in the phosphorylation, dephosphorylation. And that enzyme is, again, our friend protein kinase A. And there are other kinases, in fact, that will also do that. And one is an interesting enzyme shown right here. It's actually an AMP-mediated protein kinase. Okay? So I'd like you to focus your attention on the regulation on just this kinase. Okay? Just this kinase. So this is not cyclic AMP. This is AMP. All right? So this kinase is activated when AMP binds to it. Okay? Now, from an energy perspective, this makes a lot of sense. What's AMP an indicator of in the cell? Low energy, right? Low energy. Do we want to be synthesizing fatty acids? No. So when low energy is present, the carboxylase is converted into an inactive form by this kinase. It puts a phosphate on there. It converts the polymers back to monomers. Okay? On the other hand, once we put the phosphate on, we can always take the phosphate off. That's dephosphorylation. That's going to require a phosphatase, and there's another phosphatase that we see in the process. It takes it off, and now this guy will go back to being a polymer. Okay? So that's one aspect of its regulatory scheme. There's another one. The enzyme is also allosterically regulated. Okay? First of all, it is feedback inhibited. What do you suppose would be a feedback inhibitor? What's that? A fatty acid. So it turns out palmitate is a feedback inhibitor of this enzyme. Palmitic acid is a feedback inhibitor of this enzyme. When it's present, the enzyme shuts down. Okay? There's also an allosteric activator of the enzyme. And that is citrate. Now, let's think about this. When would we have a lot of citrate present in the cell? So if the citric acid cycle is stopped, we would have more citrate present, right? Why would the citric acid cycle be stopped? What things would cause it to stop? What's that? Well, not if oxaloacetate, but that's really not the limit. There's a more important limit. Low oxygen would be one situation. So, high energy. What if I have a lot of ATP? Let's go through the whole thing again in our heads. If I have a lot of ATP, what happens to complex 5? Stops. Complex 5 stops. When complex 5 stops, what happens to pumping of protons? Stop pumping of protons, what happens to concentration of NADH? NADH goes up. What happens when NADH goes up? Citric acid cycle stops. That's why you have citrate. There's another reason why you get fat if you don't exercise. Okay? You don't exercise, you don't burn that ATP, all these things back up, your citrate concentrations increase, and now citrate says, hey, it's time to make fatty acids. And that's what your body does. Okay? So that's a very, very important regulatory thing to keep in mind when we look at the overall picture of the synthesis of fatty acids. Okay. Well, let's get back and talk about the actual, let's see, where am I at time-wise? I've got a couple minutes. Talk about the actual synthesis. Yes, sir?
Is palmitate the only one that feedback inhibits?
Palmitate is the only one that feedback inhibits the enzyme. That's correct. And there's a reason why. Palmitate turns out, as we will see, is it's one of the major endpoints of fatty acid synthesis. And there's others, but then I'll explain that to you. Yes, sir. Do you have a question?
Palmitic acid inhibits the carboxylase?
So palmitic acid inhibits the acetyl-CoA carboxylate. That's correct. And again, that makes sense because the acetyl-CoA carboxylase is starting the synthesis of the fatty acid. So therefore, an end product of fatty acid synthesis should do that. And as we will see, palmitate is a very interesting endpoint. You'll see why that ends up in a minute. Probably not today, but probably on Wednesday. Okay? What do you guys say we just cut it out earlier today? Yeah. All right. How you doing? Oh, this was the exam, is that right?
The second one.
And
So you take it like maybe Wednesday or Thursday
and
instead of Friday.
this was because of a meeting or something, or
No, the OHSU conference, Health Careers Conference.
Oh, yeah. We need to talk about it. So just come. I'd be happy to talk to you, but do come to my office and talk to me. We can sit down and talk. Okay.
When are your office hours?
Look on my. Send me the message. I'll send you my calendar.
Okay.
Okay.
Speaking of conferences, I just wanted to check. Melissa.
I got your message.
Yeah. Melissa and I are taking it next Friday.
Yes.
So will that be all the stuff you've done
No.
next Friday,
No.
or are we going to need to watch the lectures?
It's everything. Yeah.
Okay.
So be up to that. Okay.
Okay.
Cobalt is used in a vitamin.
Cobalt is used in part of vitamin B12, huh?
B12.
Yep. You bet. How you doing?
Good. I was wondering when the start decisions were made
Start.
for.
Have you not heard? Oh, you got one.
Oh, when were they like sent out?